Hi everyone, today we're going to go over mutations and how they affect gene expression. So, just a reminder, we're going to put the mutations you may have learned about in chapter 8.5, we're going to put it in context of the central dogma, which is how DNA ultimately leads to proteins. So in this case, when we talk about mutations, we're talking about alterations in the DNA or genetic sequence, but what really it means in terms of the outcome is the mutations that occur in the DNA will ultimately affect what RNA you may produce. And if that RNA effect alters the protein you produce, that's going to result in a trait change in the outcome of what you see in the individual. So, mutations are changes in the genetic material of an organism. It could be a cell or a virus. In this case, though, the mutation is occurring in whatever genetic material they're using. It could be DNA, which is what is used by most living cells or living organisms made of cells. And in viruses, that can depend. It could be DNA, it could be RNA in the case of COVID, for instance. Now, there could be very large-scale mutations or smaller-scale mutations. So an example of a large-scale mutation is a chromosomal mutation. In this case, a large scale meaning it affects many genes at once. In this case, chromosomal mutations can be examples of loss or gains of whole or partial chromosomes that lead to different amounts of DNA compared to the typical amount of DNA found in an organism, which is known as their karyotype. So the normal karyotype of a human is about 46 chromosomes. So for most humans, they have 46 chromosomes, but for an individual with Down syndrome, they might have 47 chromosomes, or they may have 46 chromosomes, but one of their chromosomes has an extra piece or a missing piece. Oftentimes, when you have a chromosomal mutation, a large-scale mutation that affects many, many genes, this can be associated with developmental or health issues, or even death, or even the lack of development. And it's generally present at the conception of the child when, if a person has a chromosomal mutation, it's found when the cell is initially made that forms an individual when the sperm meets the egg. Point mutations, on the other hand, are what we consider small-scale mutations. In this case, because it only affects one, maybe two genes, but generally one gene at a time. This case, it alters a small number of bases in DNA, and it can be present at conception or developed in a cell later in life when a cell gets DNA damage or a replication error in its DNA. Examples of chromosomal mutations are when you might have a chunk of the chromosome missing, or you may have parts of the chromosome being added, or you may have inversion. This doesn't necessarily cause issues, except sometimes later on in more complicated processes of how sperm and eggs get made. Or you may get these weird mixtures of chromosomes, and for example, if you get this chromosome in a child, they're now missing several important genes and gained extra copies of several genes. And having extra copies of things can be okay, but sometimes it can be really damaging because the amount and dosage of a gene you get may affect the amount of proteins you make, and making too much of a protein can be damaging, such as making too much of an enzyme that breaks down bones or fat or something like that. Here's an example of a large-scale chromosomal mutation. Doesn't seem large-scale. You're only missing a little bit of chromosome 5, but in this case, you result in a condition known as Cry-Du-Chat syndrome. I like to talk about this because this is actually what my cousin has. So she has Cry-Du-Chat, and in this case, she has severe developmental delay. She's in her mid-20s now, and she basically needs lifelong care. She's nonverbal. She's about the size of a 6-year-old, and she has to be taken care of by her mom 24/7. So that's an example of just a little bit of her chromosome missing, because that little bit of a chromosome amounts to hundreds of genes missing from one of her essential chromosomes. One of the ways you can actually diagnose chromosomal mutations, you can diagnose it before the child is born. One way is it's called amniocentesis, and in this case, what they do is they draw a little bit of fluid surrounding the child. The importance is you don't poke the child with a needle. That's why you use the ultrasound. And then you can take the fluid surrounding the child. There's some cells that the baby shed in that fluid, and you can analyze the chromosomes of the child in this using this technique. So this technique has been around for a very long time, way before modern day DNA sequencing, and it's been a major way children have been able to be diagnosed with chromosomal issues early on, before they were even born. So in this case, my family knew what my cousin had before she was born, so they were able to prepare ahead of time and like have people have my uncle and aunt take care of their jobs and stuff like that, so they had more time to take care of their kid. So this is just a quick recap of chapter 8.5, but just remember, when your DNA replicates, it's replicated by DNA polymerase, which does a really good job of proofreading and preventing errors in your DNA that's made, but there still is a chance there may be an error that it misses. Moreover, the same DNA polymerase can fix damage in your cell. But again, the more damage you put in your cell, the more likely the DNA polymerase is going to miss one or two mistakes. It's really good at fixing mistakes, but every once in a while, it's going to make a mistake. And the more opportunity you give to cause issues, such as exposure to chemicals that cause damage or physical damage, or to damage to your cells, which require them to divide more often than they should, which means more replication errors, the more likely you're going to have a mutation that lingers. Mutagens are chemical or physical agents that can cause mutations, and they're known as carcinogens if they cause so much mutations in tissues that are particularly prone to becoming cancerous, then they might be classified as carcinogens. So most cancer-causing chemicals are mutagens, and vice versa. Many mutagens are often associated with increased chances of cancer. If a mutation occurs in a sperm or egg cell, it can be passed on to the child. But again, mutations, the outcome of what they do is really dependent on what actually happens. In this case, a mutation that lands in a gene, for instance, can be neutral, bad, or good if it results in a change in the protein. For example, if it changes hair color in an organism, if that organism doesn't rely on camouflage, it could do nothing. It's not a big deal. Like if you have blonde hair versus black hair as a human. On the other hand, if it results in like a missing leg, pretty big deal for the survival of an organism, especially a frog. Or it might actually create kind of slight changes in length of the body, and maybe it might help this bird get food better by increasing their beak length. It really all depends on how it gets altered, all right? Another thing to keep in mind is a mutation may land somewhere that isn't going to really do a difference in the cell. In this case, maybe it lands in a non-coding region. If it's not in a gene, it's not going to create a different protein, not a big deal. Or it may land in a gene that's not being expressed in that cell tissue. So in that event, if it's in a gene that's not being expressed, it's not a big deal. If you have a mutation that lands on your skin cells and damages a gene that affects heart proteins, not going to really do anything. Okay? And then furthermore, we'll learn about the ways genes can alter the protein if it gets into a gene, and sometimes it will affect the gene, but the resulting sequence change doesn't affect what amino acid gets coded. It all depends. So these are point mutations, mutations that are small and generally only affect a gene if it lands in a gene. Alteration of small number of bases in the DNA itself. Now, when it's a substitution, that means it's a replacement of a nucleotide. So nucleotide is neither added or missing, you're just switching it. So for example, this T is switched to a C. That would be considered a substitution. Now we may classify the outcome of those substitutions in a number of ways. It might be classified as missense, nonsense, or silent. These are all different outcomes in the protein based on what happens. On the other hand, there's another kind of mutation that's called a frame shift mutation, which is a result of either an insertion or deletion, meaning instead of switching a nucleotide, we might add a full nucleotide or remove a full nucleotide from the sequence itself. And this can have really bad results depending on where it occurs within the gene. Okay, so the ways we can classify mutations is missense, nonsense, and silence. So missense in this case You still have an amino acid where it should be, but the amino acid is different now. So in this instance here, you switch this letter from CG to TA, and the result in the mRNA is instead of a glycine, you have a serine. That's a completely different amino acid. That's missense. And that one amino acid could be a really big deal, or it could not be a big deal. It actually kind of depends on the protein that's formed. Sometimes amino acid is so similar in chemical characteristics to the original amino acid that the protein is like, it's still functioning. It's not too bad. But sometimes it can be really bad. It really depends. Okay, a nonsense mutation can be quite dangerous, and it's when it produces an earlier stop codon than where it normally occurs. So maybe the stop codon is here, and then now it's way up in the front of the protein. That means none of the amino acid, very little of the protein was formed, and now it's quite non-functional. That protein pretty much doesn't work. It depends though. Like, let's say it's a protein that's 1,000 amino acids long and the nonsense mutation occurs at the very end. That may not be a big deal, or it may still make a mostly working protein. But if it's way early in the protein, or it's like in a really essential part, that protein, not so great, not going to be happening, not going to do its job. And if it's essential protein for living and survival, very bad for the organism. It will be a very bad thing for the organism. And then finally we have silent mutations. Mutations that don't change the amino acid sequence due to the redundancy of the codon, due to the fact that some codons create the same amino acid. So in this instance here, if this C over here switches to a U in a substitution, I still have glycine. So this is classifying, and you're not going to have any alteration in what the organism looks like or how their proteins are, because there was no change in the protein. And that's it. That's a silent mutation. But just note that most substitutions usually result in missense more than any of the others, because there's 64 codons and only a few of them lead to the same amino acid, only 3 of them lead to a stop codon, and the rest lead to different amino acids. Now, an insertion or deletion is the addition or a loss of a base, and these can be very disastrous if they lead to what we call frameshift mutations. So, insertion is when you add bases to a sequence. Deletion means you lose bases from a sequence. So let me give you an example. So in this case, we're adding an A and T to the DNA one. So instead of reading my codon frames like this, by adding that A and T, instead, we are going to add the A and T here, and now we're going to read an extra codon here, and everything's going to move a little bit after that insertion. Now, as a result, I have a completely different amino acid sequence after that insertion. And this one's particularly bad because that first insertion means I don't even get 3 amino acids after that insertion. I just have a stop codon. But frame shift, this is called frame shift, where insertion or deletions alters the reading frame for the codon, and it can result in extensive missense or lead to premature stops or some combination of the two. So here's an example of extensive missense. So I have this U here. I'm going to get rid of it. It's going to be missing in the subsequent sequence. So instead of my codon frame reading this way, like it should, instead, because that letter is missing, my codon frame moves down, and everything after that kind of gets shifted as well. So in this case, I have frame shift where a bunch of subsequent, all the amino acids after that deletion get altered, and maybe even my stop codon gets lost, and I have additional amino acids that I didn't intend added to the end of my protein. Either way, my protein is way different than it was because I have different amino acids than where I originally had them. And that's going to change my protein. Now, there are examples of insertion and deletions that lead to no frame shift. So no frame shift will occur if you delete or add triplets, insert or deletion of triplets, things in threes that end up not changing the codon. So that can be a little bit confusing. So let me explain the codon reading frame. Not the codon. So here's sort of like a kind of an analogy. So imagine codons as the way you split these pile of letters into words. So the reading frame is 3 letters. So in this case, it makes a understandable sentence. The cat was mad, right? If I have a substitution, I can lead to, a word that still kind of makes sense. The cat was sad, but my triplet reading is still the same as it was. However, if I have an insertion, that's going to alter how I break down my words. So in this case, if I put an F towards the end, now I have the cat was mafada. But if it happens really early on, it's going to become very nonsensical. The faka twa sma da. So that's sort of the example of how reading frames can really mess up what codons happen after simply by adding or removing a letter. You're going to change how the rest of the quote sentence of codons are read. But if you add a triplet, like red, instead here, wherever that triplet was added, yes, you're going to have a weird amino acid that you didn't intend, but most of the surrounding protein is still intact, and you might rescue and have some decent function that was close to the original, or at least mostly like the original, may still be retained. So that's the idea of adding triplets or removing triplets. They don't necessarily change the reading frame if it maintains its codons surrounding it. So insertions, deletions don't alter the reading frame always, but if they do, we call it a frame shift. All right. I also want to point out, sometimes missense, only one amino acid change, can still produce pretty big results. Here's an example. Sickle cell disease. So in sickle cell disease here, the letters are missing from the slide. I'll go fix it on the slides. But in this case here, what is occurring is all it is I have a substitution that results in one amino acid in my hundreds of amino acids in this protein for hemoglobin in blood cells change from glutamine to valine. In this instance, though, it kind of retains some of its function. It still is able to carry oxygen and keep the cell mainly functioning as a blood cell, but unfortunately, it also makes the protein, the hemoglobin protein in the red blood cell, no longer be flexible, and instead it does this really weird chaining reaction that makes it stretch and alter its shape. So it achieves this really messed up, weird, abnormal sickle shape. And that abnormal sickle shape will actually result in the cell changing its form so that it becomes very thin and narrow, more likely to break up and more likely to clump up, which then results in a lot of the symptoms of sickle cell, where you have fatigue because you have poor blood flow, you have a higher rate of strokes because your blood cells are more likely to clot, and a lot of blood vessel damage because you're going to get backup and weirdness in your blood vessels from these clumping red blood cells. And that's an example of just a substitution leading to a little bit of missense in only one amino acid in the protein. But again, mutations, what their outcomes are, really depend on where it lands in the DNA, if it lands in coding DNA, if it lands in genes, and how it affects the amino acid and how much it affects the amino acid. Because in that instance, it affected a pretty big one, but maybe it lands in a hair cell or a hair shape gene, and that changes your hair to straight from curly. And it's not a big deal. It really all depends. So anyways, this is kind of a quick summary of all the stuff we went over. You can read over that and see if that helps you understand it a little bit better. And then this is a quick list of the words that we touched on in this lesson. All right, bye.